this video, I'm going to talk about the concept glycolysis. Glycolysis is a series of reaction which is catalyzed by various types of enzyme in which one molecule of glucose is converted into two molecule of pyruvate. In the term glycolysis, glyc means sugar, lysis means splitting. So it is a cleavage or splitting of sugar or one molecule of glucose into two molecule of pyruvate. This glycolysis is the first step of cellular aerobic respiration. Aerobic means which require oxygen. So though it is the first step of aerobic respiration, glycolysis does not require oxygen. In any step of glycolysis, it does not require oxygen. Where does the glycolysis takes place? Glycolysis takes place in the cytoplasm of all cell of all organisms. So glycolysis takes place in all organism which is present on the earth. Glycolysis is also called as EMP pathway. It is called EMP pathway because it is discovered, or the scheme of the glycolysis is given by the three scientists, that is Emden, Meyerhoff and Parnas. The first letter of each scientist together named EMP pathway. So EMP pathway named after the three scientists who gave the scheme of glycolysis. Now let me discuss about the actual steps of glycolysis. Glycolysis mainly has two stages, that is energy requiring stage and the second stage is energy yielding stage. Each stage, that is energy requiring as well as energy yielding stage, has each 5 steps. So 5 plus 5, 10 steps. In total, glycolysis have 10 steps. Now let me talk about each steps of glycolysis. In the first step, to the 6-carbon compound, that is glucose, a phosphate group is added, and this phosphate group is from the ATP. So phosphate group from the ATP is added to 6th carbon of the glucose. That changes glucose into glucose 6-phosphate. So here 6 refers to the 6th carbon to which phosphate group is added, and that phosphate group is from the ATP. So ATP is utilized in this step, means it is a energy requiring stage. So ATP, that is adenosine triphosphate consisting of 3 phosphate, is converted into ADP, that is adenosine diphosphate, because one phosphate is utilized by the glucose and it convert that into glucose 6-phosphate. That is the first step, and this first step required, or it is catalyzed by the enzyme known as phosphorylase or hexokinase. That is the first step. In the second step, glucose 6-phosphate is converted into fructose 6-phosphate, and this is because of change in the position of the atom or because of the internal arrangement or isomerization. So because of the isomerization, glucose 6-phosphate is converted into fructose 6-phosphate, and this step is catalyzed by the enzyme known as isomerase. So that is the second step. In the third step, one more phosphate group is added to the fructose 6-phosphate, and that phosphate group is again from ATP. So one more ATP is utilized in the third step, that is it is the energy requiring stage. So one more phosphate from the ATP is added to first carbon of the fructose 6-phosphate. That changes fructose 6-phosphate into fructose 1,6-bisphosphate. So here 1,6 refers to the position of the carbon to which phosphate is added. So 2-phosphate is added, that's why it is bisphosphate. So fructose 1,6-bisphosphate. So here ATP, that is adenosine triphosphate, is converted or changes into adenosine diphosphate or ADP, and this step is catalyzed by again the enzyme known as phosphorylase or hexokinase. That is the fourth step. In the fifth step, the fructose 1,6-bisphosphate is split or cleaved into 2,3-carbon compound. Fructose 1,6-bisphosphate is a 6-carbon compound. This 6-carbon compound is split or cleaved into 2,3-carbon compound, that is glyceraldehyde 3-phosphate and dihydroxyacetone phosphate. So this is the actual lysis or splitting of the glycolysis. Glycolysis means splitting or cleavage of the glucose or sugar molecule. So actual cleavage or lysis takes place at the fifth step where the 6-carbon fructose 1,6-bisphosphate is cleaved into 2,3-carbon compound, and this step is catalyzed by the enzyme known as aldolase. So 2,3-carbon compound is formed, that is glyceraldehyde 3-phosphate and dihydroxyacetone phosphate, and these 2,3-carbon compounds are the isomers, means they can be interchangeable, which means dihydroxyacetone phosphate can be converted into glyceraldehyde 3-phosphate, and this interchanging process is catalyzed by the enzyme known as isomerase. So finally 2 molecule of glyceraldehyde 3-phosphate is formed. This 2 molecule of glyceraldehyde 3-phosphate undergo several changes to produce 2 molecule of pyruvate. That's how 1 molecule of glucose is converted into 2 molecule of pyruvate. Anyhow, only one reaction is shown here. That's why I have written 2 into in each step. So here 2 into means 2 molecule is produced in the each step or at the each step. In the next step, the inorganic phosphate is added to glyceraldehyde 3-phosphate. You have to note here, this inorganic phosphate is not from ATP. From some inorganic sources, inorganic phosphate is added to glyceraldehyde 3-phosphate that convert glyceraldehyde 3-phosphate into 1,3-bisphosphoglyceraldehyde. So 1,3-bisphosphoglyceraldehyde is an aldehyde. This 1,3-bisphosphoglyceraldehyde later get oxidized or undergo oxidation, and for this oxidation it requires an oxidized form of coenzyme like NAD. So in this step NAD plus is changed or reduced to NADH and H plus. So NAD plus is reduced to NADH and H plus, that converts 1,3-bisphosphoglyceraldehyde into 1,3-bisphosphoglyceric acid. So in this step glyceraldehyde 3-phosphate is converted into 1,3-bisphosphoglyceric acid, and this reaction is catalyzed by the enzyme known as glyceraldehyde 3-phosphate dehydrogenase. So from glyceraldehyde 3-phosphate 1,3-bisphosphoglyceric acid is formed. In the next step, this 1,3-bisphosphoglyceric acid will split and the phosphate group is removed from the first carbon of this compound. So, the phosphate group from the first carbon of 1,3-bisphosphoglyceric acid is removed and it is transferred to ADP, that is adenosine diphosphate. So, ADP is converted into ATP, that is adenosine triphosphate. The phosphate group from the first carbon of 1,3-bisphosphoglyceric acid is transferred to ADP, that changes into ATP. So, one molecule of ATP is produced here. So, this step is energy yielding stage because one ATP is synthesized here, that converts 1,3-bisphosphoglyceric acid into 3-phosphoglyceric acid, and this reaction is catalyzed by the enzyme known as phosphoglycerate kinase. In the next step, this 3-phosphoglyceric acid, which consisting of phosphate group at the third position, this phosphate group from the third carbon is transferred or shifted to second carbon of this compound. So from the third carbon, the phosphate group is shifted to second carbon, which convert the 3-phosphoglyceric acid into 2-phosphoglyceric acid or 2-phosphoglycerate, and the enzyme which catalyzes this reaction is called as phosphoglycerate mutase. In the next step, from this 2-phosphoglyceric acid, a molecule of water is removed. From each molecule of 2-phosphoglyceric acid, a molecule of water is removed, which convert 2-phosphoglycerate into phosphoenol pyruvate, and the enzyme which catalyzes this reaction is known as enolase. Next is the last step where phosphoenol pyruvate will broken or it splits and the phosphate is removed from the phosphoenol pyruvate. This removed phosphate from the phosphoenol pyruvate is transferred to ADP, that is adenosine diphosphate. Upon receiving phosphate, this adenosine diphosphate is converted into adenosine triphosphate or ATP. So in this step, ATP is synthesized. That's why it is the energy yielding stage. So ADP is converted into ATP upon receiving phosphate from phosphoenol pyruvate. Thereby, this phosphoenol pyruvate is converted into pyruvate, and the enzyme that catalyzes its reaction is known as pyruvate kinase. So ultimately, two molecule of pyruvate is formed from one molecule of glucose. So that is about the different steps of glycolysis, where one molecule of glucose is converted into two molecule of pyruvate. I hope this video will be useful. If you like this video, please hit the like button and subscribe to my channel.